Hello, my name's James Beastie. I'm here with Charlie Power and Charitha Senaratne. We're from Langton Grammar School for Boys, and we're here to talk about the Mylin Basic Protein Project, MBP2. Every nerve cell in the central nervous system has an axon which is surrounded by the myelin sheath. This is composed mostly of lipids and proteins. The most abundant protein in the myelin sheath is called proteolipids protein 1, which we'll come back to later in the talk. The second most abundant protein is called MBP. This is thought to have a role in maintaining the integrity of the myelin sheath by cross-linking the layers and acting like a sort of glue. The myelin sheath electrically insulates the axon, protects it from damage, and minimises the amount of energy required to transmit impulses by up to 1/2000th of the amount. On the left here you have a neuron with a healthy myelin sheath, and on the right, a neuron with a damaged myelin sheath. Damage to the myelin sheath can result in reduced nerve function. On the right shows an MRI scan of a patient suffering from multiple sclerosis. As you can see at several points, you have these white spots. These are called plaques or scleroses, which is where the myelin sheath has been damaged and nerve impulses are no longer being transmitted. If you have several of these plaques or scleroses, you have multiple sclerosis. What causes multiple sclerosis? There are several factors in the development of multiple sclerosis. One is a genetic predisposition. For example, of the 100,000 sufferers in the United Kingdom, 3/4 of these are female. Another example is if you look at some of the Scottish Islands, whereas the national average for multiple sclerosis is 1 in 600, in some of the Scottish Islands you're looking at 1 in 100 are suffering from multiple sclerosis, and this is often blamed on the inheritance of their Viking genes. Another potential cause is the lack of vitamin D. Around the equator, where there's a lot of sunlight, the frequency of multiple sclerosis is very low. However, as you move further away from the equator, levels of sunlight decreasing and levels of vitamin D decrease, frequency of multiple sclerosis again increases. Exposure to viral infection can also trigger development of the disease. For example, Epstein-Barr virus, glandular fever, can trigger development. Another factor is age. It's the most common neurological disease in young people and is usually diagnosed in the early 20s and 30s. MS is a very serious condition. Here are just a selection of the effects and symptoms. There are three main types of multiple sclerosis. The most common is shown on at the bottom here, relapsing remitting multiple sclerosis. This is where you have a series of unpredictable attacks in which your condition gets worsens very quickly. Then you have a period of remission where you recover slightly and you regain some functionality. You can then go weeks, months, or even years before your next attack. As I said, it's unpredictable. The second most common is called secondary progressive multiple sclerosis. This is shown on the graph at the top. This is where you start off much like relapsing remitting multiple sclerosis with a series of unpredictable attacks followed by some remissions. However, when you reach a certain point, your condition continuously deteriorates and there are no attacks, it just continuously deteriorates and there's no breaks. The least most common and the most serious form of multiple sclerosis is the one in the middle, primary progressive. This is where from the moment you develop the condition, your condition continuously deteriorates and there is no recovery. What is MBP2? MBP2 has been running for 8 years. We're funded entirely by the Wellcome Trust and are supported by the University of Kent. We have scientists, students and academics from the School of Biosciences coming and working with us. Students at the Langton carry out original research into MBP, which, as I said, is implicated in the development of multiple sclerosis. MBP is a very fragile protein. There is no tertiary structure, there are no ionic bonds, there are no covalent bonds. There is only secondary structure, alpha helices, beta-pleated sheets, and the hydrogen bonds within. It's also subject to a variety of post-translational modifications, that is to say, changes to the protein that happen once it has become synthesized. Specifically, we're looking at phosphorylation of certain amino acids, threonine, tyrosine, and serine. There is evidence to suggest that a changed shape of myelin basic protein can trigger an autoimmune response where white blood cells cross the blood-brain barrier and start attacking the myelin sheath. This diagram shows a cross-section of the myelin sheath. You can see in between the layers you have Mylin Basic Protein. How does this work? If you look at this area here, this is where one specific amino acid, threonine 98, is located. The phosphorylation of threonine 98, we currently believe that increases the bond angle here, which pushes these arms further apart, which means that the layers of the myelin sheath, which are normally very tightly packed together, can become a lot looser, which can be what triggers an autoimmune response. As you can see, PLP1 is also a transmembrane protein, which Chiruka will talk about later in his talk. I'll now hand over to Charlie, who will talk more about what we're doing currently in MBP squared.
I'll try and make this quick. Okay. Oh, wait, no. Okay, the first thing we had to do was clone our MBP gene. We alter it slightly and then insert it into the expression plasmid. We encode it and use it PCR, polymerase chain reaction. We also designed primers, which will sit at either end of our gene. These primers include specific restriction enzymes, which are engineered into the sequence, along with a run of DNA, which will code for 8 histone amino acids, which will be added to the C-terminal end of our protein. Okay. To next. Oh, yeah, no. Okay. Wait, I need to. Okay. Sorry. A bit nervous. Okay. Sorry about this. The PCR reaction essentially fills in the gaps between the primers just to synthesize your target. To start your target DNA. Multiple rounds of this reaction yield large amounts of DNA. This technique is used by forensic scientists to amplify DNA in scenes of crime. So now I got it. Here you can see wait, how do you even use this? Oh, there. In the pink here is our MBP with the cut size Hindi3 and Echo R1. These are shuttle vectors, which mean it can work in E. coli and yeast. We also have a GAL promoter here, which means that when our yeast cells are, when the plasmid is in our yeast cells, means that we can change the medium it is into one containing glucose into one containing galactose. This means this switches on the promoter, leading to the synthesis of MBP mRNA to MB, which will be translated into MBP protein. The point of this is that the biochemistry of yeast is very similar to the biochemistry of humans. We are using yeast as our model organism in the expectation that it will treat the MBP in very much the same way that a human cell might. Go to our next one. Our next step is then to purify the MBP. We do this by using nickel cephalose chromatography. This involves coating the cephalose beads with nickel sulfate. If you remember, during our PCR cloning, we added a run of 8 histone residues. We did this because histone binds to nickel, meaning that when we pass our yeast lysate, if you can see here, through, only some proteins will bind, which is our MBP. No other yeast proteins will bind. So then we can just wash off the yeast proteins, and then we're just left alone with our MBP. Okay, and here. So here you can see our MBP and how, no, wrong one. Basically, our MBP is purer and larger than commercial MBP. This is because it has 8 extra amino acids. How can we identify its MBP? We run a normal SDS page gel, transfer the proteins onto a membrane, which is essentially a sheet of paper. Then we have to block the protein, because the protein can, the membrane is basically designed to bind to any protein. And as we are using antibodies, our antibody will bind to any on our protein. So then we have to use a non-specific protein, which will basically cover anywhere that does not already have our protein on it. We use a primary antibody, which will only bind to its specific target protein. But however, this primary antibody has no way of showing us where it is, so we have to wash the blot and use a secondary antibody. Fun. Yeah. Okay, so here you can see our yellow. This is our primary antibody, and the red is our protein, our MBP. And we have to use a secondary antibody, but it has to have a reporter enzyme attached, and we use horseradish peroxidase. The advantage is this, that several secondary antibodies will bind to the one primary antibody, giving us a degree of amplification of our final stage. Our final stage requires the addition of 4-chloronanthol and hydrogen peroxide, which will leave a black precipitate in the resulting reaction, showing us where the reaction has taken place. So yeah, I'll give it to Teresa.
So on the left here, you can see the original SDS Page gel that we ran, and it's stained blue with Kinase stain. There are 3 tracks of interest. This is the track for the wild-type human MBP, then a mutated form of MBP which we produced, and then bovine MBP. So the middle panel shows a Western blot that we ran using anti-MBP as our primary antibody, and this confirms the identity of the protein we produced as being MBP. And you can see from these 2 tracks here that we produced roughly equal amounts of wild-type MBP and our mutated form of MBP. The panel on the right here shows another Western blot where the primary antibody was not anti-MBP but anti-phosphothreonine 98. This is an antibody which only binds to MBP that has been phosphorylated on threonine 98. You can see that there are signals from the wild-type MBP and from the bovine MBP only. This is because the wild-type MBP has been phosphorylated by yeast kinases, and the bovine MBP has been phosphorylated in the cow from which it was sourced. However, there is no signal from our mutated form of MBP, and this is because we use site-directed mutagenesis to change threonine 98 to alanine 98, and alanine cannot be phosphorylated. Therefore, despite this protein being MBP, it's not showing up on that blot. One of the major advantages of using yeast as our model organism is that there's a knockout library of 6,000 non-essential genes which have been individually knocked out of the yeast genome to produce deficient strains. We identified 2 kinase deficient mutants, YK1 and MCK1, and 2 phosphatase deficient mutants, PPT1 and PPZ1, kinases being the enzymes which phosphorylate and phosphatases being the enzymes which dephosphorylate. We transformed the SLBS1 plasmid, which contains the gene for the wild-type human MBP, into our various strains, and then grew up our transformants and induced MBP. We then obtained a cell lysate and ran Western blots of the protein from those lysates. Unfortunately, we came into a problem here because our method of visualizing the Western blots using 4-chloronaphtol wasn't quite sensitive enough. So Nadia from the University of Kent, whom we met earlier, kindly took the samples up to the university and ran them there using ECL, enhanced chemiluminescence, and visualized the blots. On this slide, you can see our results. So the blot on the left has the primary antibody as anti-MBP, and the blot on the right has the antibody as anti-phosphothreonine 98. On each blot, the tracks are representative of wild-type human MBP, MBP from the YK1 strain, which was kinase deficient, a kinase deficient mutant, MBP from the MCK1 strain, which was also a kinase deficient mutant, and then MBP from the PPT1 strain, which was a phosphatase deficient mutant. Unfortunately, we couldn't get results from the PPZ1 strain because we are unable to obtain a cell lysate. When yeast have non-essential genes knocked out of the genome, they still have to find alternative pathways for functions of the cell to occur. In the case of PPZ1, this meant that the strain was sickly to start with, and we think the additional stress of having to try to express our MBP protein killed that strain. We can look into shorter induction times in the future, but for now, we have no cell lysate. So you can see from the left-hand Western blot that in all the strains shown, MBP was expressed, though to varying degrees. The reason you're seeing this different intensity is because though the total protein load for each track was the same, the proportion of that total load, which was MBP, differed depending on how much the particular strain expressed the MBP. You can see that the pattern for relative expression is the same for the two pretty much the same for the two blots. And this is suggestive that the kinases, which are on phosphatases, which are deficient in these three strains here, are not the ones responsible for phosphorylating or dephosphorylating MBP in yeast. So what's the next stage for MBP project? So the most abundant protein in the myelin sheath is proteolipid protein 1, PLP1. PLP1 is a transmembrane protein that has 4 transmembrane domains, and it's known to associate with MBP in the myelin sheath. The role of MBP, as stated earlier, is thought to be holding different phospholipid bilayers together, and we're interested in investigating the potential interaction of PLP1 with various mutated forms of MBP. To go about this, we would obtain MBP, as per usual, on our nickel-cephorous column. At this point, MBP is denatured due to the methods used. However, because MBP is a protein that only has secondary structure held together by hydrogen bonds, if we allow it to, it should spontaneously reform those hydrogen bonds and so regain its structure and be renatured on the column. We can then present it with some PLP1 to see if it binds and what happens. The one problem with this approach is that PLP1, being a transmembrane protein, will not be in its native state outside of a membrane. However, there are ways around this, and we will deal with this when we get to this stage of the project. Thank you for listening. I hope you have
Questions? One or two.
So you might have said this already, but what was the connection with your Mylin Basic Protein and multiple sclerosis? Has there been any mutants been found or any defects in the protein found in patients?
So the role of MBP is holding the different parts of the phospholipid bilayers in the myelin sheath together. And if you imagine, I wonder if I can get back to the, no I can't. So there was a diagram shown in the part of the presentation that James did where you can see that the MBP protein is like a sideways U, and there's a curved part of that which is a molecular switch region. And if that part is modified, say by phosphorylation, the charge will change the shape of, oh, it's back. It will change the shape of the MBP. So if you can see where the pointer is, if that curve, if there's phosphorylation here, for example, that will change the shape of the MBP, and that forces the arms apart. And that means the different layers of the myelin sheath come apart. And what brings about MS is an autoimmune response. So when those myelin sheath layers come apart, then it becomes easier for there to be attacks on the myelin sheath, which finally break it down. So we're interested in looking at the different post-translational modifications which can cause this.
Oh, you've already answered my question, actually. But, someone else.
It's not really a question, but it's potentially another exciting opportunity. We've just got Wellcome Trust funding, actually, to make genetically engineered regulatory T cells targeted against MBP, which we'll be testing in mouse models of MS. We've just started that project, so we should maybe collaborate, have a cross-school collaboration.
Excellent. Okay, thank you. I think we've probably better call it a day there. Thank you very much, guys. Well done. Thank you.